Economist, one of the most prestigious publications in the world, September 30th, 2023. That's 3 weeks after last year's RadFest. They dedicated an entire issue to the search for living forever. And they had one article after another telling us pretty much what you know already about delaying aging, but they let the rest of the world know. Some of the higher end people on this planet reading The Economist were realizing we can live a lot longer if we behave differently. And then the Bloomberg Business Week, and again, a very high end publication read by some of the wealthiest people in the world, people who may donate some money to accelerate research. They talked about the prospect of living forever, this immortality obsessed frontier that we are all engaged in. It's making it into the mainstream media. Now, we've talked about George Church since 2016, his work with CRISPR-Cas9 gene editing therapy. Back in 2016, a lot of people were critical. Can you really do this and make something work? Well, FDA approved the first CRISPR-Cas9 therapy December 2023. Everything I'm showing you is after last year's RadFest. Then a month later, FDA cleared another CRISPR therapy to treat thalassemia. This is an anemia that a lot of people suffer, and this can now be genetically treated in a way that the person will maintain normal hemoglobin. This is a major breakthrough, and Wall Street Journal, recognizing all this, talking about doctors can now edit genes inside your body. And again, you're going to hear a lot of people like Liz Parrish and others talking about gene therapy and how that can be used even today to reverse many, many aspects of biological aging. New England Journal of Medicine, well, this is the Dean of Harvard talking about the new era of gene editing in medicine. When we started in 2016 talking about gene editing, it was still kind of science fiction. It's now moved into clinical reality. People are going to start to be treated with gene therapies to treat age-related illnesses. The Saudis, they're trying to spend $1 billion a year on research, but identifying scientists with the credentials, the competence, the skill to do some of these research projects hard. But they are spending money right now, which is great news. They've got a lot to spend. If they see a project that needs money, they often will write a check and fund that sort of thing. And of course, the commercial companies raising, in this case, $300 million to figure out better ways to reprogram old cells back into young cells and enable people to potentially rejuvenate any damaged organ, potentially enabling us to live for a very long time, if not forever. Now, we're going to talk a lot about epigenetic age here for my presentation, and many other scientists will talk about it. And this is a way to identify, looking at DNA methylation patterns, what the risk of age-related disease is, and frankly, how long you're going to live. So the epigenetic age, it measures DNA methylation. It can tell you your biological age, and if you're going to engage in an intervention, some experimental therapy, well, you want to get a baseline epigenetic age test, and then after you're done with the therapy, we'll follow up and see how much younger it may have made you. So this is considered the most accurate way of assessing whether people are aging faster or slower, and we sure don't want to be aging faster. And the good news is there are 2 different companies in Rad City offering this test this weekend. You can have your blood drawn and have this test done and find out how old you are from a biological standpoint. So longer lived people, well, they have a younger epigenetic age. And if you have an older epigenetic age, it may mean you need to more aggressively deal with your overall health, because older epigenetic age, it's associated with lower levels of physical and cognitive functionality, and it reduces your longevity. So this is a very important test, and the good news is companies are offering it. So what happened in early 2024? Well, exosome-rich young plasma, and this was published finally in old rats, it cut their epigenetic age in half. That research is now advancing to dogs. The inside story I'm hearing is it's working as well as in dogs as it did in rats. A study we helped fund, young plasma reversed epigenetic aging in old rats, increased their median lifespan, the human equivalent of about 7 years. That's with young plasma. And then OSK, Yamanaka transcription factors, reversed epigenetic age, improved overall health, extended remaining lifespan. And we're evolving that research, our research group, into monkeys, as are some other research groups, which is good news, and we're going to try to make those old monkeys grow younger, and we want to replicate everything in rat mice models when we can, move into monkeys when we can, and you're going to hear some people talking about potential clinical research being done with OSK right now being done. You're going to hear about that at RadFest. So I'm going to explore that exosome-rich plasma study because it's really important to understand. This was given every other day for 8 days, and it cut old age in half. This is an unprecedented finding published February 2024. And what's impressive about this is the dosing regimen is practical. It was given every other day for 8 days. And who wouldn't want to go into a doctor's office every other day for 8 days, have exosome-rich young plasma put in your body, and cut your epigenetic age in half? I don't think anyone in this room would not do that. And the idea is make it affordable so everyone can make sure they benefit from it. But to explore this study more, they were able to reverse the epigenetic age in the liver, 74%. And I think of so many people who have died of liver failure, this therapy could save their life. In blood, 64%. And that hints they were potentially rejuvenating the hematopoietic stem cell niche in bone marrow. And if you do that, well, you've got better immunity, you deal with anemia, start producing more platelets, red blood cells, and the heart, a 46% reduction in epigenetic age. That could save people from congestive heart failure. There's millions of them in the United States. Hundreds of thousands die. This one therapy could eliminate heart failure as a cause of death. In the brain, about a 24% reversal of epigenetic age. Now, these are unprecedented findings, and these charts, I'm going to make sure everyone in this room clearly understands what they mean. The red is the control group. They're aging normally. They have a high epigenetic age. They're older. The blue that you see there are the control animals, the control young rats, and they have a young epigenetic age. But that orange is what should impress you. These were old rats given the young plasma. And in the blood, their epigenetic age returned almost to that of a younger animal. And the same was true with the liver. The reversal of epigenetic age back down to that of a younger animal. These are life-saving therapies. In the heart, reversal of the epigenetic age of the heart back down to that of a younger animal. And again, the red are the control group. They're aging normally. We don't want to do that. The blue are the young animals. We wish we could just stay young. But with this therapy, we can potentially put this into our bodies, and we are going to be experimenting with this therapy on monkeys to see if we can't reverse aging in monkeys the way it was done in rats. And again, the brain, about a 24% reduction in epigenetic age. It's the hypothalamus. We want to do way better than that, and I'm going to show you some slides on how we're going to do that. Another measure of epigenetic age, the tissue of the old rats. Again, that red is where we don't want to be, but it happens to be where most of us are, and i.e. we're aging, and it's reflected in our epigenetic age score. The blue, the control rats, the young ones, wow, if we just stay young, it'd be fantastic. But with the exosome-rich young plasma, the old rats start to resemble that of the younger animals. That's in the blood, that shows it in the liver, shows it in the heart, shows it in the brain again. Two different ways of measuring epigenetic age, both showing aging going in reverse. But what about conventional markers of health, like liver function? We get older, and you see those red dots? Those are old rats whose liver function is worsening. This is something that happens to people, especially if they're obese, non-alcoholic fatty liver disease or alcoholic fatty liver disease. Either one, you want to do something about it. And then the younger rat, it's got healthier liver function. But look what happens when you introduce exosome-rich young plasma into old rats. Their liver function on all these 4 tests, look at that, it is improved. It goes back down to that of a younger animal. Older animals given exosome-rich plasma have their liver function markers restored to that of a younger animal. And conventional cardiovascular risks, triglycerides, they go up as we get older, and we don't really want that to happen. The young rats have lower triglycerides, but when you put that young plasma into the old rats, their triglyceride levels go down. And in fact, what you're going to continue to see here is if we can use young plasma and some of these gene therapies, you won't have to swallow so many pills. Your aging process is going to reverse in a normal way, and you're going to stay healthy. HDL beneficially increases in response to the young plasma. Cholesterol decreases. And what's most exciting, glucose. So many of us have glucose control issues. And I say that in the context, ideally, you want your glucose level, your fasting glucose level, to be under 85 milligrams per deciliter. That's ideal. It's very challenging as we get older. We lose insulin sensitivity, and we try to target a little bit below 100. And some of us can't even do that, even with anti-diabetic medications. In this case, exosome-rich plasma moved that glucose level back down to that of a younger animal. And then kidney function. So many people suffering chronic kidney disease. Creatinine levels going up in the old rats, just like it does in so many older people. We don't want that to happen. The young rat, of course, has lower creatinine levels, so its kidney function is better. But look at that. The old rats given the young plasma, their creatinine levels goes back down to that of a younger animal. This could be a potential treatment for people with chronic kidney disease, so they don't have to go on dialysis, don't have to go on a waiting list for a transplant. It's absolutely miraculous. These three measures of kidney function all improved in response to young plasma. And I guess as a caveat, I should let people know, I don't sell young plasma, just so you understand. I'm letting you know what the research findings are. We're going to study these in primates to see if we can't accomplish the same types of results as was done in these older rats. So to put it succinctly, young plasma, old rats, saw their epigenetic age in blood, liver, heart reduced to that comparable with younger rats. This is a major, major advance. And the exosomes, for those who are not familiar with them, they're secreted by our 37 trillion cells, and they enable these cells to communicate. Just imagine if we didn't have exosomes being secreted all the time to have our body work in a systemically effective way. Well, we wouldn't survive. So exosomes, they're secreted by hematopoietic stem cells to boost our immune function, our blood volumes, and then mesenchymal stem cells, and anti-inflammatory properties, and tissue repair. We like healthy exosomes in our body, and that's what our mission is with the old monkeys. Put lots of healthy exosomes into their body to enable them to live longer. Now, this study was published this year, and what it enabled us to do in concert with interacting with the scientists themselves is identify how many exosomes does a 150-pound person need to emulate the rat study, where aging, biological epigenetic age went in reverse. Well, we need about 4.8 trillion exosomes in each treatment. Every other day, young plasma plus 4.8 trillion exosomes, and that would emulate the rat study. But before people do this, we suggest let us do it in primates. Let's see how well it works in older primates to see if we can reverse that. But to put this into context, some people are undergoing umbilical cord plasma infusions. And they're good, except they only contain a few 100 million exosomes. And this study showed that 4.8 trillion, a lot more, produced those incredible, incredible results. For those who want to read more about this, you can go to our age reversal website. You can read these studies and to see if you have a neurodegenerative condition. There's a lot of evidence out there that young plasma might help. And anyone considering that, please undergo some blood tests first and undergo the treatment and do a follow-up. See if what's being published in literature applies to you. The more evidence we're able to gather, the more confident we'll be in recommending people do that. I'm going to take a water break and play a little commercial for you that I created. It was a little bit different than the original.
Today we celebrate the first glorious anniversary of the Information Purification Directives. We have created, for the first time in all history, a garden of pure ideology, where each worker may bloom, secure from the pests of any contradictory thoughts. Our unification of thoughts is more powerful a weapon than any fleet or army on earth. We are one people, with one will, one resolve, one cause. Our enemies shall talk themselves to death, and we will bury them with their own confusion. We shall prevail.
I had a lot of fun making that, and I think you know what side we're on there. So this is another young plasma study that our group helped to fund. We do fund millions of dollars a year in biomedical research. Almost none of it has anything to do with making money. We want to figure out how we can live longer. In this case, we simply used young rat plasma, put this into older rats, and we get some really interesting findings. This is a charitable group that we fund that does this research. We have a number of charities that we support to do research that has no commercial applicability but could result in advancing the science. So what we did here, beginning at the human equivalent of about 60 to 65 years, every other week, we had the rats injected with young plasma, not exosome-rich plasma, just young plasma. The control group didn't get any treatment. And a survival curve over a period of 26 to 30 months, it declined, of course, in the rats that are not giving the treatment. If you're over 60 to 65, people start dying off. But the treatment group, the young plasma treatment group, none of them died over that period of 26 to 30 months. None of the young plasma animals died. Now, this is median lifespan. We're not talking about maximum lifespan, but we at least showed something that if you're programmed right now to start developing some fatal diseases, the young plasma might delay that. And this chart demonstrates that. The gray dots on the left are the control group, and their median lifespan is reduced. And you can see them dying. Every time you see them dropping, that means more and more of them are dying off. The young plasma group in red, they're living longer. They're not dying at the same rate as the control group. And then on the right, you see some photos, and you see a control rat there looking older, and the treated rat remaining looking younger. We're seeing this consistently with the research. We don't expect it. We're just looking to see how long can we make these animals live, and how healthy are they, but they also look younger. So the good news is when we start to reverse aging, you might start to look younger, and I think a lot of people want that. But the DNA methylation in these older rats in response to young plasma every other week, it improved, and we were able to see improved insulin sensitivity, better immune function, and reduced inflammation. Huge problem with aging. We have chronic inflammatory reactions that destroy our brain, our kidneys, and we just start to age to death. But here's the interesting caveat from this study we funded. Remember, we extended median lifespan 9%, the human equivalent of about 7 years. But the longest lived rat in this study was in the control group. And that rat happened to have very good genes. It let it live longer. It didn't even need the young plasma. It just continued to live. And what does that tell us? Well, we need to reprogram our genes so that we emulate the longest lived people in the world, the longest lived animals. And if we do this in such a way, and I'm going to describe it in just a minute, using OSK Yamanaka factors, we could theoretically reprogram our old cells back into younger cells and have them regenerate for an indefinite period of time. Transcription factors are proteins that help turn on and off specific genes. And if you put in the right transcription factors, you can reprogram old cells back into stem cells or younger cells to continually regenerate tissues throughout our body. So we're going to talk about now Yamanaka transcription factors. These were discovered in 2006, and there are 4 of them: OCT4, SOX2, KLF4, and c-MYC. The acronym is OSKM. You're going to see that acronym used throughout presentations this weekend. You're also going to see it in the popular media because the media is starting to pick up on the fact that this transcription factor combination is able to restore youthful functionality to old cells. And what we're doing with our research is we're only using 3 out of 4 of these Yamanaka factors, the OCT4, SOX2, KLF4, and we're seeing better results with 3 out of 4 as opposed to using all of them. So I'm going to try to depict a little bit about how we're achieving the delivery of OSK into the cells. That's very, very important. So in this case, all 4 of them, and many studies have used all 4 of them being delivered into old cells. And one option is to have your old cells removed and then have them reprogrammed with those Yamanaka factors so they convert back into a pluripotent stem cell, and then that can differentiate into healthy young cells to help regenerate your body systemically. But that's a burdensome way of accomplishing something. So our newer way of approaching this is we're not going to use the c-MYC anymore. That stimulates, by the way, excess cellular propagation. This is great for an embryo, but for older people, we may not need that. Just 3 out of 4. The OSK embedded into old cells, and then allow those old cells to partially reprogram back into a younger cell. And then we don't want to just let that go. We want to maintain the youthful cells via intermittent expression of OSK. Deliver that OSK into the cells and turn it on and turn it off so we achieve an optimal amount of cellular rejuvenation. So we get you from an 80-year-old back down to a 30-year-old. And if you're happy with that, we just keep you at a 30-year-old simply by delivering more OSK into your cells as the cells you use it up. So what happens when you introduce these transcription factors into old mice? Well, you learned 2 years ago, it reverses aging systemically. The Salk Institute people published this 2022. I talked about it at RADFest. It made international media headlines. The idea, for the first time, aging systemically reversed. Old mice growing younger again, a major, major breakthrough. And what happens with partial cell reprogramming is, well, it can work in vivo, in whole live mice, and these transcription factors did not produce toxicity, did not increase cancer rates. They did what we intended them to do. And again, I mentioned younger looking skin. This is what we're seeing in response to these rejuvenation therapies. People look younger. Inflammation is decreased, reduced senescent associated secretory phenotypes. We're reducing the senescent cell burden in response to these therapies. Now, those studies were done 2022. Look at the date of this study, 2024, in which aging was again reversed by a group of brilliant scientists that we are interacting with right now. And what they were able to do, cyclic induction of the OSK expression in old animals. These were about 77-year-old human equivalent. And they were able to increase remaining median lifespan 100%. That's huge. Now, that's not the whole life of the animal, it's its remaining lifespan. But that's important for all of us in this room because we're older. If something only worked in young animals but failed to work in older animals, we'd be rather depressed. Good news is this worked. It improved the health of the older animals in relationship to control mice, profound age reversal in the heart and the liver, assessed again by those DNA methylation epigenetic age clocks, and they were even able to show some petri dish reversal of human skin in the petri dish. In fact, there is some patents that are going to be granted very soon, not by us, but they're OSK topical creams. They seem to reverse aging of the skin back down to whatever age you want. This study, using just the OSK, it was able to improve the frailty index score in a significant way. And I think we all know as we grow older, we become frail, we become more vulnerable to falls, fractures, and premature death. So the frailty index score improved. This is a validated measure of aging, and OSK was responsible for doing this. And again, the prospect, and I've seen videos of this, and it's shocking, where the OSK is applied to an old man's skin, only one hand. And after a couple weeks, couple months, that OSK skin treatment shows very much younger skin. So there's a potential that we get to live longer and look younger again. So the challenge is identifying the best way of delivering these Yamanaka factors inside of cells. Now, the way that's typically done is using a viral vector. And this technology, I hope you're as impressed with it as I am. You're talking about a virus which is 150 times smaller than a typical human cell. It's very tiny, but the science has emerged and has advanced to the point they can embed OSK into a virus and then have the body infected with that virus. This is an innocuous virus, and in doing so, you deliver OSK into the nucleus and into the DNA. So the viral vector is what's proven, and this is what we'll be doing first with the monkeys, but there's also plasmid vectors that people are looking into, and we're very excited about lipid nanoparticles in which we could control precisely how much OSK gets into your body, including your brain. The brain loves to suck up these lipid nanoparticles, and if we attach OSK to them, we could theoretically induce systemic rejuvenation. And then chemical reprogramming. Bear in mind, OSK, these are normal. They're in our embryo. They express themselves when we're younger. They keep our bodies a lot younger, and then we lose them with age. So the idea of chemically reprogramming old cells to start naturally producing OSK may be a different way to induce the expression inside our cells. So we had this remarkable extension in lifespan. And again, these mice were the equivalent of 77-year-old people, and we were able to reprogram their cells, increase their lifespan, improve frailty scores. And as far as the lifespan. Control mice, they died after 8.86 weeks, but the ones given the OSK, they lived over 18 weeks. But here's what should shock you more than anything else that I might present here. It turned out, the scientists who did this, by the way, brilliant pioneers, and when you're a pioneer, you don't do everything perfect. But it turned out that they only delivered a fraction of the intended dose of the OSK into the bodies of these older animals. They wanted to deliver a lot more. They only delivered a fraction of it, and they still achieved the remarkable results I just showed you today. And to remind you, the OSK-treated mice living 18 weeks compared to only, and that's like a 10-year benefit for humans. So control mice had a remaining lifespan of about 5 human years equivalent, and then the OSK-treated mice, their remaining lifespan equated to over 10 human years. And if we can double your remaining lifespan, meaning instead of dying in 5 years, you are facing death at 10, well, at that point in time, we may have achieved indefinitely extended healthy lifespans, and then you don't have to die. And again, even if the animals don't live significant longer, they did live longer in better health. So we both extended remaining lifespan, we being the scientific group that we are working with, and they were able to enable them to live longer in better health. So you look at what happened in early 2024, and wow, more happened, by the way, in the last 12 months, and anyone who thinks differently should let me know. More has happened than everything that has occurred in the past. There's never been a conference where this type of information was able to be disseminated, because this kind of information didn't exist back a couple years ago. It's now published, it's now respected, and we are moving this rapidly into old primates. We want to see old monkeys grow young again, and you're going to hear some people this weekend talking about using it in themselves. I'm not advocating that, but there are people who are doing that. So another study that we helped to fund, and this was a rather crude way of introducing these into the body, but we injected OSKM into the hippocampal region of the brain, and what we found was, well, a lot of benefits occurring as far as memory and learning improvements in response to OSKM, literally in a syringe through the skull of the old rats, and we were able to rejuvenate their brains. And the hippocampal region, rather important. We may notice that our memories and our learning abilities aren't the same at our age as where they were when we were younger. Well, these improved in a rather dramatic way. So what you're looking there, a 36% reduction in latency time, which is good. It's a good measurement. It takes the old control rat a rather long time to figure out how to get out of a maze. But you treat that old rat with the OSKM, and again, a crude way of treating them, a needle right into the brain, but no, it didn't hurt the animals, by the way. You can see it's much better. It's much better. And this is another way of measuring this. And I don't want to get people's hopes up too high as it relates to the results of the study, because while we improved the learning ability, the red are the control group, the blue are the old OSKM. But the real fortunate ones are the ones that are still young. You can see that they're really doing much, much better. So we haven't achieved total reversal yet, but we're working on it very hard. This, again, is a different graph showing the memory performance. The young mouse is doing really good. The old control are very bad, but the old animal given the OSKM, and that's injected directly into the hippocampal region of the brain, it's improved. It's improved about 30% compared to that old control animal. So the history of OSK and OSKM transcription factors. 2006, Shinya Yamanaka showed that old cells can be reprogrammed back into youthful cells. 2011, old human centenarian cells reprogrammed back into an embryonic stage. 2022, aging partially reversed systemically. Whole body age reversal in a couple independent studies. 2024, we're moving to rejuvenate old monkeys, and as you're going to see on stage this weekend, old humans are doing some self-experimentation that we're not involved in, but nonetheless, we want to follow what they're doing to see how well it works. And please understand, before 2006, this was all considered impossible. Nobody in the biology field who was conventional ever thought this could be done, and it turns out it was done. Now, I was invited to attend a private seminar. J.P. Morgan Chase, one of the largest wealth management companies in the world, they're losing their customers because they're getting old and dying. So they put on this seminar, brought in some experts from the Buck Institute to teach wealthy old people how to evaluate longevity data, see what kind of lifestyle changes can improve their life. And what they were able to analogize was your body being a depreciating asset. Okay? I mean, getting old, you're depreciating. Maybe put some effort into changing your lifestyle. And what they said was 93% of your lifestyle determined by your choices, your lifestyle choices. That's 93% of your lifespan determined by lifestyle choices, about 7% by the genes you're born with. So think of it, longevity in investing terms. Make the right choices to extend your healthy lifespan. And if you do most of what they say the best lifestyle choices are, they say you can live to be 25 to or 15 to 25 years longer.